All right. So this is the next chapter, which will tie in with chapter 3, and it's particularly the cell membrane. And this is how everything gets into and out of the cell. So we talked about plasma membrane being a phospholipid bilayer. It's got the glycerol heads and fatty acid tails, or phosphate heads and acid tails. And so it's able to regulate to some degree what comes in. And you should be looking at the picture in your book on page, for those that are here, I'll project it on the screen. Caden, would you pull that screen down, please? Just as long as it's as low as that board on the wall, it's fine. A little bit more, yep. Thank you. Okay, so anyway, that picture in your book, guys, is on page 68, and I'll put it like, for those of you that are here, I'll put it up on the screen so you can look at it. But no. Anyway, it's much more than just a membrane. I mean, that's what we think of it as just the part. But there are other parts in that allow more versatility within the plasma membrane. So it makes a boundary, keeps the inside in and the outside out. And that's important. It's important to have that barrier. It's important to have that barrier that's able to facilitate movement to and out of the cell, get rid of waste and get nutrients in, oxygen in, and carbon dioxide out. In the case of respiring organisms or organisms, including our cells. It can't just be random as far as what goes in and out. It has to be specific to what those materials are and what the cell needs. And the diagram of that is something that's called the fluid mosaic model. So from art, guys, are you familiar with what a mosaic is? What is that, Wayne? What's a mosaic? A bunch of pieces put together. That's exactly right. So if you look at the picture here on the screen, again, those of you that are viewing the video, it's on 68 in your book. It's made of a bunch of different pieces. And if you look over there, the pieces are proteins sticking out, the phospholipid bilayer, there are cholesterol molecules that are embedded in there. And so it's just as Lane said, it's made of pieces. Okay. Those proteins that are in the boundary or in the barrier, in the membrane, can either be peripheral or they can be integral. And if they're peripheral, it means that they stick out. It means that they stick out on the side, and if they're integral, it means that they go all through. Now, if you look at that picture again on the screen here, and I'll show it to you here. This protein, it says, is integral. And the reason to say this protein is integral is if you took that protein out of the membrane, what would happen, KK? Remove this protein from the membrane, what would it leave? It's going to leave a hole. It goes all the way through. So now the membrane's not rating at all what goes down. Things can just free move in and out. That protein is integral to the actual continuum that is the membrane. On the other hand, this sticky outy one right here, what's it say it's called, Kenny? Look over on the screen. This one. It's peripheral. And when we think of peripheral, we usually think of our vision. What does peripheral vision mean? Out to the side, right? So that's why I call it a peripheral protein, because it just sticks out to the side. If Brenna, if we remove that peripheral protein, would it make a hole all the way to the membrane? Difference in the one that Caden was looking at, the integral one, it goes clear across. So if you take it out, it's going to leave a hole. If you took the peripheral one out, it'd leave a hole in one side of it, but not all through. And so an example of that would be thinking about a vehicle and being integral, vital to its function and not vital to its function. Let's think about drive shaft of a vehicle and the front fender of a vehicle. So which thing would be integral to the operation of a car, the drive shaft or the front fender? Because, is your car going anywhere without it?
No.
Could your car still drive? Didn't have a front fender? Yeah. So front fender would be peripheral. It's just kind of on the outside side of the car, but it's not critical function. The drive shaft, without it, you've got no car. It's not going anywhere. Okay. So think about that as being akin to what we're looking at with the protein. Okay. There are carbohydrate chains that attach to the lipids, and those are called glycolipids. And then there are also ones that attach to the proteins, and those are called glycoproteins. So I'll put this back up for those of you that are here, but for those of you that are watching, what we're talking about there is this right here. This chain of green. And look at them. I'll back over there in a moment. Green hexagons. Why suppose these carbohydrate chains are made of green hexagons? How many sides does a hexagon have? 6. Does anybody remember what the monomers of carbohydrates are? You remember that from chapter, yes. Remember the graphic organizer? What were the monomers of carbohydrates? Monomers were saccharide. You remember the examples of them? Yeah. Glucose, fructose, and galactose. So glucose, fructose, and galactose are all isomers. That is, they are the formula C6H12O6. So Lane told us that green hexagon would have 6 sides, and that represents the 6 carbons, monosaccharide. So that chain of green hexagons represent carbohydrate, or a sugary part that's attached. The one on the left, it's attached to the red ball, so it's attached to the lipids. One in the middle that's attached to the blue blob, you might say, it's attached to a protein. So now it's called a glycoprotein. And it says that those glycoportions, those sugary portions, are used in cell recognition. What we mean by that is, it's not like, I meet Kenny in the hallway and I say, "Hey, I know you," right? It's not that recognition. It's not a visual recognition, because cells and molecules aren't capable of that. But the recognition comes in the fact that, again, could it fit together like this, right? Do you remember talking about that a number of times? It either fits or it doesn't. So it's the example here of ABO blood types. Okay, so let's say that I had surgery or something, and Aaliyah went and donated a unit of blood, and they gave me that unit. Would it matter what her blood type is, or my blood type is? That would work well or not, Bennett? Okay, it does matter, right? We have to be not the same blood type, but it has to be compatible, or it won't work. So you can follow this. So when we talk about A and B antigens on a blood cell, we're saying there are some kind of projections like these that stick off of those red cells. So let's say that my wrist is the red cell, and my index finger now, if I look at it like my index finger represents an A antigen, my pinky finger represents B antigen, and so blood cells look like this. What type of blood would I have? What did I say this was? A, and this is, and so if both on my red cells, I would be type AB. What if I don't have either of them? Then I'm type O. If I just have this one, I'm type A, and if I just have this one, I'm type B, right? So everybody's blood cells, depending on what antigens stick out from them, they're going to be recognized differently. So if I have type O and Aaliyah's blood has A or B, how is my body going to react? It won't go well at Kenny, because I'm going to recognize the blood she donated for me as being foreign. And so my antibodies are going to attack those red cells, and it can cause some different things, like kind of clog up your kidney tools, I think, because it's attacking those. But as long as, if I have AB blood, what kind of blood could I receive? A and B and O. Because remember, O doesn't have anything. So that's why we say O is the universal donor, because it looks like this. Is anyone's blood going to recognize this as form? Well, no, because it doesn't have anything to recognize. Okay? If I'm type AB, I have this. So look, guys, if I already have this one and this one, I could get this one, or I could get this one, or I could get this one, or I could get this one, because I already have all of those things. Okay. You don't have any assignment to do for that. Just get started. We're just getting started on the chapter. And again, we won't spend very much time on this, but I would like to do the lab. And so you guys, I think, will be in the lab probably tomorrow. Okay? You're